I'm going to talk to you today about how we can actually develop and understand how we can actually get better formulations using cutting-edge drug delivery technologies for the next generation of nutraceuticals health formulations. Just in terms of my own background, I'm not actually a director of saanroo. I'm actually a director for a centre. So I'm a professor of a university in Australia. I head a centre for pharmaceutical innovation. So you'll see from my experience, I'm a drug delivery guy, really. Just in terms of my own interests, you'll see there's a bit of a crossover here. So obviously today, the interest is the intestine is a biological barrier. It's an important recognition that how do we get these small molecules, biologicals, into the bloodstream to do their job. It's all about the technologies we can use there. But I also am very interested in infections and even cancer nanomedicines. And there's nice, interesting crossover there because obviously we were just talking about the microbiome. So understanding the relationship, and you'll see from my talk, there's some great connections there. Oh, here's some of my team in Australia. I'm sure they're all working hard, or I think it's some evening currently. I mean, just in terms of, spruiking my own background, we've got a nice centre of about 150 people, and we've got excellent capabilities across the pharmaceutical innovation space, but it also translates into the nutraceutical advanced formulation space. So we can take new chemistry through new materials, through preclinical into clinical studies. I've got some rather nice labs. So anyone ever in Australia, come and look me up. I'll show you a few shiny labs. So today, what do we talk about? We want to talk about leading longevity ingredients. So as a pharmaceutical scientist, when you first think of an ingredient or an active, look at its structure. Because in the area of trying to maximize bioavailability, and we've heard that enormously across many talks, and everyone's making claims around bioavailability, understanding your molecule first is essential. And I'll show you how certain structure, certain properties of certain active molecules lead to different routes of formulation and delivery, challenges and opportunities. And then, of course, I'll talk to you about the gut as a biological barrier. What do we need to do to maximize absorption, to maximize bioavailability? And then we'll talk about the various delivery technologies, different types of materials. What are they, again, opportunities and challenges? Opportunities are you can massively improve the absorption of actives. Challenges are there could be some toxic effects, or there could be some other unknown interactions in the body. And then, of course, we've just had an amazing talk there around, or a workshop there around the microbiome. So obviously the gut health axis is critical. And I'll show you how this can play an interesting role in the way that longevity ingredients can obviously directly be active in the gut. But we'll also show you that the formulation itself can play an important role there, both potentially positive and negative. Give you some case studies from my industry partners here. And maybe we can have a chat about where the future's going in this space. So anyway, as a pharmaceutical scientist, what do? We look at molecules. Now, hopefully many of you maybe you haven't all done chemistry, but these are, at least you'd know these actives. We can't go wrong. Vitamin C, omega-3s, curcumin, berberine. And of course, we've been talking earlier about collagen and its breakdown products, the various peptides. So there's a whole range of molecules there. And when you look at a molecule, as me as a chemist and a pharmaceutical chemist, I first of all look at the molecule and say, is this molecule going to be charged? How does its structure influence its solubility? What sort of formulation is going to be ideal? Is it going to like lipid? Is it going to not like lipid? That will play a role in how we best formulate things. And there are obviously two major challenges in bioavailability enhancement. One is around solubility and one's around permeability. And of course, peptides and proteins, you don't get large molecules going across the gut very easily. So that's a permeability challenge. And we'll cover some of those interests. So it comes down to this is like pharmaceutics 101. There are these various types of categories of molecules, particularly in the drug area. But the ideal is class 1, a molecule that's highly soluble, highly permeable, great. Easy to formulate. You can just go in there as a powder or a solution, no problem. But of course, the other molecules have got those either solubility or permeability, or both. So obviously the class 4 molecules are the hardest. You have to overcome both the solubility and permeability. And I'll talk to you a little bit about that. But of course, some other important challenges. When you look at a molecule, you might say things, is this molecule stable? What's its metabolism? Will it be metabolized pre-systemic, or will it be metabolized within the body? So molecules can sometimes, when we take them orally, they can be actually metabolized within the gut, or which can be good or bad. Sometimes the metabolite is the active, sometimes the metabolite is inactive. So understanding that, and maybe also using encapsulation delivery systems to control that, can be quite interesting. So, the gut. I think most of us here may not be all biologists, anatomists, but we know the gut. We know very interesting biological feature. The stomach, clearly low pH, small intestine, large intestine. The majority of absorption happens in the small intestine, sometimes some in the stomach. But we know also the microbiome is in the large, well, there's microbiome everywhere, but the major microbiome region is in the colon, and that has that interesting interplay between, but there's some very complex areas around the gut, around things like gastric emptying. Certain formulations, certain times of taking certain nutraceuticals can actually have a very, very different uptake depending on the time, the conditions, and there's lots of good science around this. And there's also this interesting thing around driving. You may also be able to, when drugs do pass across the intestine, or nutraceuticals, they can go via the lymphatic transport mechanism as well as the normal portal vein. And targeting lymphatics can be quite interesting. It can have a number of interesting opportunities for bypassing the liver and therefore changing the metabolism of certain molecules. So it's good to understand. And we've been talking a lot about personalized medicine. And I think this is some very data that's probably not well received. Men and women are different. And their gut and their various biological features of the gut are different. So we look at these, this is a lot of data here, but there's just about every feature of the gut, whether it's gastric emptying, whether it's the mucosal associated bacteria, whether it's the activity of various molecules. Women and men are different. So this is an area I think at the forefront. We're talking about personalized medicines. If you're trying to maximize the absorption of a very interesting bioactive, there's a great potential here to make women formulations, men formulations that may have a very different, we know there's difference in dose just based around average body mass, but around knowledge of the absorption processes, I think this is a future frontier for us to think about going forward. Anyway, back to the oral transit and absorption. There's a thing called the pharmaceutical food effect that we may or may not know about, but this is a list here of some well-known pharmaceutical ingredients, and you measure, they've got low solubility, which many actives have, but they've typically got a 3 to 5-fold increased absorption in the fed state. So you may know when you take many medicines, it says, restricted compliance, take this with food or take this without food. It's quite interesting. I've worked on one particular prostate cancer drug, and when you take it in the fed state, it can absorb 10 times more. So that means, and in fact, Johnson & Johnson got that drug approved, and the variability in the fed state was so high, they had to get it approved in the fasted state, which is actually a bit counterintuitive, get it approved for use. So I think in this area, I'm not seeing a lot of recommendations, but quite clearly, if you know your molecule has a very, very high food effect, and you can use your formulation to drive that, and that's what I'll be talking about, but you can also put it on the packaging. If you want to get higher bioavailability of certain lipophilic actives that are poorly soluble, take it with food. And of course, back to these types of ingredients. So there's two sort of general types. A molecule that's poorly soluble, which many of those actives are. It can either be the more hydrophobic, brick dust, so it doesn't like lipid, or it can be the lipophilic, a grease ball type molecule, and of course that opens up lipid-based drug delivery technologies, simple lipid emulsions, but I'll talk more about that. So this gives you the opportunity of thinking about, if you've got a brick dust type molecule, you probably wouldn't go down the same route of formulation. So what are some of the opportunities in formulation? Well, first of all, physical modification. You can reduce the particle size if you've got a solid active, which you don't want to put lots of different excipients, by literally nano-sizing it, you'll get a higher surface area, you'll drive solubility kinetics, and you'll improve absorption. You can do things like create a bit of chemical modification. You can form prodrugs, or you can form salts. And I've seen a couple of people, groupings here, that are making some co-crystal technologies where you can actually take a molecule, create this interesting co-crystal that has a higher solubility and improves absorption. But of course, the thing that I'm most interested in, so my industry partners, are reformulate into a carrier. That can be a colloidal or sort of a nano or a non-nano, a micro or a nano, and they have their advantages and disadvantages. They can be polymeric in nature. They can be inorganic. People do use the things like silica and other things like clays as inorganic carriers, or probably most commonly, they're lipid-based formulations. And I'll just give you a little bit of, when you're thinking about drug delivery generally, or nutraceutical delivery, what are some of the requirements? Well, obviously you want to have your materials to be biocompatible. It's essential. In this industry, you've got to have food grade materials as your carrier. So you've got to have low toxicity, and ideally they degrade and don't accumulate in the body, of course. So you've got to have that. Sometimes you can design your carrier with a controlled release. Sometimes you might be interested in actually specific extracellular targets that you can actually build into your carrier. You want to cross biological barriers. You want to keep maximize your cell-drug interaction, whether that's in the body or actually when there's the drug's in the blood system and it's actually interacting with various components of the body. And then obviously you want to have the drug carrier has to have integrity. You don't want it. So there's no point creating a nice encapsulation delivery system that falls apart very quickly when you dose it. That would be counterproductive. So there's another. So let me move on to sort of lipid-based drug delivery systems. I'm sure you've all. The simplest is take a liquid lipid, put some a nutraceutical that's lipid soluble in it. That will create an opportunity for enhanced absorption. But of course, we can make more complex and more advanced. So lipid emulsions, micelles or microemulsions, self-emulsifying drug delivery systems. What they are is you've usually got a lipid component, but you build in some co-solvents and often some surfactant in there as well. So those are clear, homogeneous liquids that once they get put in biological relevant media, into our gastrointestinal tract or into aqueous media, they disperse into very, very small nanocarriers or microemulsions or nanoemulsions. We can also actually, in the lipid space, use solid lipids. And they're quite interesting. So there's solid lipid micro and nanoparticles. And what they enable us, probably give us a more controlled release system. Some of those first systems there, they are what you call immediate release. They transport the drug across the gut quite quickly. But some of these solid lipid carriers are quite interesting. So obviously, and then you move on to the more exquisite lipid complexes. You can actually sometimes complex a molecule with a lipid, particularly if they're different charge. A cationic molecule, a lipid with an anionic nutraceutical can create a complex that can also drive absorption. Then you move into the mesophase system. So a liposome is a lipid bilayer. But there are some even more advanced systems. I like working with these things called cubosomes. You think of a liposome, it's just literally like a lipid nano bubble with a single phospholipid membrane. If you screw it up into 3 dimensions, you can create these little things called cubosomes. And they can be great for increasing the transport of both lipophilic and water-soluble compounds within their nice internal structures here. And of course, many of us have had the COVID vaccines, which have been relied on these lipid nanoparticles. So we know that the mRNA vaccines that were fast-tracked through the COVID pandemic, they use this technology, which is actually a quite complex nanocarrier, which has very important cationic lipids that complex the DNA or the RNA into that 100 nanometer particle. But that's obviously for injectable. But some of these systems can also be used in different application modes. And another thing, a thing that I'm very interested in is making hybrid lipid delivery systems. So we take some of those advantages of a lipid, then maybe we incorporate it with another carrier, a solid carrier. Maybe it's an inorganic, or maybe it's a prebiotic. So you might imagine you can actually build a system. You can take some of these formulations, maybe liquid, which doesn't always have the ideal end application. And sometimes it's nice to have tablets and powders to put in capsules, because sometimes a liquid has challenges in terms of shelf life, etc. But we can actually create these interesting hybrid systems using those technologies. So why lipid-based drug delivery? Well, generally non-toxic, biocompatible. There's a protective action you can create. We can build in, engineer encapsulation. It facilitates biological barrier crossing, which is an important thing. It can mimic biology. And then we can use this fatty food effect to maximize solubilization of poorly water solubles. We can reduce metabolism, efflux things. We can increase membrane permeability. We can stimulate intestinal lymphatic transport, which is something that's quite interesting. And we can protect against proteases. I think in one of the previous talks, talking about collagen, the first thing that happens when a biologic goes into our gut, proteases digested. When we eat meat, the large proteins do not go across our gastrointestinal tract. It's the peptides, maybe small polypeptides, that are produced through the peptase action. But then, what are some of the challenges of lipid-based drug delivery systems? Well, maybe for certain molecules, you might not get very high loading levels. So if you've got an active that you want to dose at many gram levels, you wouldn't want to dilute it by putting it into a formulation that's got a low loading level. There are ways of getting around that. For some systems where you only need, the tens, hundreds of milligrams, that may not be a challenge. Physical instability. We know things like, remember the old-fashioned days where we used to have real milk? Remember when milk came out of a cow and you used to get this thing called cream? That was because, the milk emulsion is unstable. Whereas, obviously, nowadays, our milk is homogenized and it's got surfactant in there. But physical, and then also chemical stability. And we know there's a lot of interest in iron delivery, various iron-based formulations. Iron being a redox molecule, it can actually have an interaction with particularly unsaturated lipids and cause oxidation, and that can cause shelf life challenges. So you need to know about these things. Following on from the last talks, the gut health axis, you can't miss it. We've spoken previously about the brain, the heart, the skin, oral health, liver, immune system, hormone. We know it's exploding in terms of our knowledge of how this can actually be. So first of all, our active molecules, many of the active molecules that I'm going to talk about, do have a positive action in the microbiome. And that can be done in two ways, of course. It can be done through systemic. So the molecule goes into the blood system, then it can have its action within the gut. Or for molecules that don't pass through, so obviously prebiotics, they generally don't absorb those fiber type molecules. They just literally go into the microbiome region and they create short-chain fatty acids, anti-inflammatory properties, which invigorate the microbiome. But knowing more about this is quite important, and what are the opportunities and risks for longevity ingredients around the microbiome. So I mentioned many ingredients do actually boost the microbiome, and formulation excipients, some formulation excipients actually have a negative effect. We've just published a paper on a very well-known medicine that's out there, and we've shown that the actual ingredients that are used to facilitate a peptide absorbing, they're actually very surfactant-like molecules that actually open up the junctions of the small intestine to get peptide drugs into the blood system. When you actually look at their effect on the microbiome, it's highly negative. So that's an interesting thing, is that I think the whole understanding of how our formulation excipients play either a negative or a positive role in the microbiome is something we haven't discussed enough. So there's potential to design your drug delivery or nutraceutical delivery system with a better microbiome outcome, and there can be a very, very interesting interplay. I nearly asked the question in the last talk, what's the future? We've shown in some of our work that some molecules, their absorption is very different when the microbiome is either in a healthy or a non-healthy state. So there is, back to our talk about the various gut health axes, there is actually a gut axis. So most of this stuff is in the colon, but the feedback into the small intestine and various transporters can actually, we've seen situations where the bioavailability of a certain antipsychotic molecule was increased several times when we gave prebiotics to the gut. So obviously there's opportunities there. Now I want to move on to some, my amazing partners, Saanroo and Pharmaco. They are world leaders in high quality formulation. So we're talking about the whole nutraceutical industry. I think what's really important, if you're going to make next generation high quality products, you've got to have well-designed technologies that are in some way validated with respect to understanding how they work, the quality control, the quality of the ingredients, and of course, using the right technology for the right active, because one size does not fit all. So here, this is an array of their technologies. An Aquasil is a self-emulsifying drug delivery system, which is great for things like omega-3s. And we've published clinical trial data showing improvement there with Amanda, who's in the audience there. Lipisperse is a cold water dispersant powder. So one of the things you've obviously seen, things like curcumin. If you just put curcumin in water, it does not disperse. It does not have much solubility. There are ways and means of creating nice formulations where you keep the drug in a molecular format. Compressible powders. So obviously you can make interesting, you can use carrier molecules to create, what it does, it stabilizes the molecule into a molecular form, and then it can be used to make tablets. And then when it goes into the body, it disperses well. It can be designed to disperse, and it releases the drug in this molecular form, which maximizes the transport. And liposomes. I think we can look around everywhere. You see people are using liposomes. Unfortunately, many people are not using well-characterized liposomes. To make high-quality liposomes that are either unilamellar, 100 nanometers in size, which may give you the ideal performance, you have to basically know what you're doing. And the plexosome technology that Pharmaco and Saanroo market is a high-quality example of that, and used for a range of actives. So I'm just going to finish off with a few case studies where we can see some examples. So I just mentioned earlier, curcumin. This is a bit of a little image here. So if you put curcumin in water, it sits on the top. And if you can imagine how much actually solubilizes, the oral bioavailability of powdered curcumin is very low. Obviously, here we have the hydrocurc technology, which uses this thing called lipisperse, which enables it to get great dispersion, and that correlates into human clinical outcomes. So the data here is a bit small, but you might get this presentation later. This is human clinical data showing the pharmacokinetics. Many people don't show. You often see people claiming bioavailability enhancement so many times, but to do high quality, powered human clinical data and get significant enhancements. And here you've got several times enhancement. So, it's the area under a curve in a pharmacokinetic study is important. But actually, one thing drug delivery or one thing delivery technologies can do to your nutraceutical, it can also change the half-life. So many actives, if you can control the absorption, you can also keep the active in the therapeutic window for longer. And that's a very important thing. So if you, some molecules will have a very short half-life, and there's a certain concentration where they are doing their job in our body. They're doing their antioxidation, they're overcoming various disease states. But understanding that and understanding the whole pharmacokinetic profile is very important. Moving on. Plexosome, I mentioned that a little bit earlier. This is a nice little schematic. This is really a drug delivery type thing. In drug delivery, people use liposomes because you can actually do things like create targeting. You can put molecules that they are used for things like cancer therapy. But the plexosome technology is a high quality liposome which has been well characterized. It's been shown with high-end techniques, things like transmission electron microscopy, that you've got monodispersed, sub 100 nanometer particles with a nice lipid bilayer, and there's a range of actives there that have been formulated, and there's lots of evidence around human PK data and also preclinical and in vitro data to show how these molecules work. So a plexosome technology, a liposome, it can do two things. It can improve the solubility, but it can also improve the permeability. And I'd probably say the permeability is something that's very nice, because if you think of the gut, and I showed you that image of the gut's epithelial layer, you want to get molecules to transport across those enterocytes in the gut. And by the definition of these fuse beautifully. Liposomes, if they're made well, they fuse with biological membranes, and they transport molecules across them. So that's why they're interesting. And these particular ones have been scientifically validated. They're high quality ingredients. They're made under GMP. They're made in Australia under license in a therapeutic goods Australia. So, but of course, you will find many liposomal formulations that probably won't be optimized and will not give the same performance. So I think in this whole area, there's potential for better regulation. And it's something I think we need to move forward with, so we actually have quality control. Otherwise, we're going to be, giving our population poor quality nutraceuticals, and it's not going to work. Moving on to some of the saanroo products. So Levagen. So PEA, there's the molecule down there. That's one of the molecules I showed. It's a well-known active, and it's a challenging molecule in terms of its normal bioavailability is low. So but it's an important endogenous fatty acid, amine, used with pain, inflammation, stress. So there's the saanroo have done many large clinical trials where they've shown both bioavailability enhancement, but also many efficacy outcomes. So there's been 15 published studies around this formulation, and I should show a little bit of this just shows this is the raw active ingredient. This is where you've got it in this the lipidisperse technology, which enables massive improvements of dispersion, and then that correlates with solubility, and then that correlates with both absorption into the bloodstream, better bioavailability, better outcomes. The final I'm going to talk about this OEA, another well-known bioavailable active. Similar story in terms of, this is the body's way of being like a natural GLP-1. It obviously is an endogenous fatty acid amine. It's synthesized in the small intestine. It can actually regulate appetite. So it has got those interesting anti-obesity, gut-related benefits. Again, powered by this lipidisperse technology. High quality formulation that does what it should, and there's been obviously lots of outcomes, lots of published work on this. So obviously, by using the right technology for the right molecule, you can get the optimum outcome. That can lead to much more efficient products, better quality products, less waste. And of course, sometimes you have your drug absorbing, you don't want it to go down the toilet. This is from my own research. I mentioned earlier the idea of maybe creating hybrid lipid formulations where we can build in some interesting performance. So we know dietary fibers. We've heard in the previous talk things like inulin and others are great at invigorating our microbiome. We can also use it to create some microcapsules. So here we've made some rather nice microcapsules containing liquid lipid in the core, and we've created in this one here, an inulin shell. And we've chosen the inulin for the right molecular weight and the right properties. And we've been able to show that actually this technology, not only does it encapsulate lipophilic actives, it maximizes absorption, and it improves the gut health. So it's in a single formulation, it's a sort of and there's also a very interesting interplay between the gut health and the absorption. So it's one of those situations, and I think the whole future of optimal formulations is by coupling ingredients. So here we're coupling the excipient and so we're using two excipients, both with interesting properties. One is enabling absorption of the drug, one's invigorating the microbiome, and then we've got our active that can do something combined. So I think the future here, we can have a situation where 1 + 1 equals 3, or 1 + 1 + 1 can equal 5, if you understand the various mechanisms going on in various technologies. So hopefully I'm getting a bit on time or not on time, but I'm going to finish off. So thanks for your patience. Hopefully you like my mad publication presentation style. So, but hopefully I have shown you a little bit around, if you're thinking you've got an active nutraceutical, you should know a little bit about its molecular structure. What are the challenges? Is it likely to be poorly soluble? Is it likely to be poorly permeable? So let's make a formulation that's going to overcome those barriers. Because obviously quality is what we want. We want extremely high quality formulations that will go into the populations. And you need to have that bit of knowledge around the molecule and the formulation. And I think I've shown you briefly that the gut health axis is an amazing target for longevity ingredients. Many actives do actually have that action, but if we maximize their absorption bioavailability, and maybe if we also couple them with a formulation that also benefits the outcomes, we can probably have a next generation of outcomes in this study. So thanks very much. Thanks to Pharmaco and saanroo for bringing me along to tell you about these technologies, and please come and see these guys at 5:54, I believe, not very far away. So come and talk about the amazing technologies that are on the shelf here. And there are a number of pre-packages, but these technologies can be transferred into a large number of other bioactives that you may be interested. Thanks for your attention. Happy to answer any questions or